Hey, A&P Peers, this is Dr. Jones checking in. This is A&P 2, Lecture Test 6, Video 9? I think we're on 9. Yep, 9 works. Video 9, and we're starting babies. We're talking about development. We've talked about sex, now we're talking about babies. So video 9, we're starting on the top of page 15, Chapter 29, Development and Inheritance. Dun, dun, dun. All right. So development is fertilization to maturity. So this is the growing up. Prenatal development happens before birth, so that will include both embryonic development and fetal development, and we'll define those terms here in a second. Postnatal development is after birth, so from birth to maturity. Maturity here is skeletal maturity when you stop growing taller, and that's what we're calling maturity. Maturity is when you stop growing taller. When your physical growth is completed. There are other forms of maturity that are harder to measure, of course. If only we could measure everything with a right-hand x-ray. But this is effectively when the growth plates fuse. So when those epiphyseal plates become epiphyseal lines, you are done growing taller, you are mature by skeletal definitions. Senescence is age. Fertilization/conception is when the oocyte and the sperm fuse and make a zygote. Pre-embryonic development, and again this is new in the new version of the book. If you have the previous version of the book, you don't have these terms. So I'll tell you, pre-embryonic development is the first 2 weeks of development post-fertilization. Did it move? Oh, okay, it's the same picture. I got confused. Okay, so embryo is weeks 2 through 9. Second week through the 9th week. Embryonic development is the development of the embryo, so development from weeks 2 through 9. Embryology is the study of embryonic development, which is the changes between weeks 2 and 9. And the fetus is 9 weeks to birth. And then fetal development is the development of the fetus between weeks 9 and birth. And bottom line, if it's the size and if it's a clump of cells, it's a pre-embryo. If it's the size and shape of a cashew, it's an embryo. And if it looks remotely human/alien, then we're going with fetus. We said that. Differentiation is when you form the different types of cells. Differentiation is when you go from a single-celled zygote to lots of different types of cells, liver cells, bone cells, skin cells, hair cells. All of these different cells include the same DNA. And so turning some of that DNA on and some of it off and specializing is called differentiation. Inheritance/heredity are your genetically determined characteristics. The easy ones to see will be things like height, skin color, eye color, hair color, and then there are other ones that are harder to see, like lactose intolerance type stuff. And then genetics is the study of inheritance/heredity. Dun, dun, dun. Okay. Fertilization involves the fusion of 2 haploid gametes. And in the previous chapter, chapter 28, reproduction, we made gametes, right? We talked about mitosis to make the sperm cells, and then meiosis to divide into gametes. And the male makes 4 sperm per stem cell, and the female makes 1 giant oocyte. The sperm are just genetics and mitochondria in a tail, effectively, enzymes. We'll talk about that. And then the oocyte, which is released as a secondary oocyte, is the genetic material, enzymes, and all of the cytoplasm for the entire human being to be made. And then these extra little bits of genetic material that are unused and left over are the polar bodies. There can be 2 polar bodies, or if the first polar body undergoes meiosis II, then there are 3. So that's why. Fertilization involves the fusion of 2 haploid gametes, back to the note set, the oocyte and sperm, each containing 23 chromosomes, producing a zygote that contains 46 chromosomes. I have a picture of that. There we go. A zygote that contains 46 chromosomes, which is the normal complement in a somatic cell. And a somatic cell are the body cells. All the cells in your body are somatic cells, except for the sex cells, which are the eggs or sperm that you make. I guess there are people who make both. That's rare, though. Very, very rare. Fertilization generally occurs within a day after ovulation, 24 hours, sometimes 48 hours, although please recall that sperm can be kept alive in the female reproductive tract by the PEG cells for up to a week. And here you can see the size differential between an egg and the sperm. There's a huge size differential. Okay. Page 16. The sperm delivers the paternal chromosomes, and the oocyte delivers the maternal chromosomes and, of course, all of the organelles and cytosol. Before a sperm is capable of fertilization, let's see, is this typed in the next slide? I don't think so. It must be exposed to seminal gland and PEG cell secretions, so those are the 2 things needed to cause capacitation in the sperm, both seminal gland and the PEG cell secretion. So the male and female reproductive tracts both need to be functioning correctly for fertilization to occur. We're top of the page? Okay. Of the nearly 200 to 250 million sperm per ejaculation, only 10,000 enter the uterine tube and fewer than 100 reach the oocyte. Oh, that looks like more, but functionally sterile is less than 20 million sperm per milliliter. Now be careful here because they're going back and forth between the how many million sperm per ejaculation and how many million sperm per milliliter. So standard is 200, I think the previous book said 200 and the new book says 250 million per ejaculation. But both editions of the book agree that less than 20 million sperm per milliliter is functionally sterile because while that's a lot of sperm, it's not enough to actually reach the oocyte. Why can a single sperm not fertilize an oocyte by itself? The sperm, as you recall, on the top tip, the outside of the front of the head, is surrounded by an acrosomal cap, which is one of your lab terms, if you don't know that. I still am putting cards on each of these. They should show up in a corner somewhere. I think they show up over here. And I put them at the 1 minute mark, generally. So if you go to the 1 minute mark on this video and you're on the right device, because the cards don't show on all devices, then there should be a link to the playlist for Lab 4. Okay, if not, and you're seeing this somewhere other than YouTube, I should be searchable on YouTube as Dr. Jones 32, which is the same way I'm searchable under Quizlet, which is Dr. Jones 32. So, if that helps. Okay, so the acrosomal cap, which is a term we need for a lab, and now for lecture. And then need dozens of acrosomal caps worth of enzymes to break down the corona radiata. So here's your oocyte with the zona pellucida surrounded by a crown of cells called the corona radiata, which is a crown that radiates around the oocyte, corona radiata. Okay, list some of the enzymes and their functions. I swear I have this typed in somewhere. Yeah, I don't have it typed in here. Okay. So the enzyme, first enzyme is hyaluronidase, H-Y-A-L-U-R-O-N-I-D-A-S-E. O-N, hyaluronidase. I hate writing with a mouse, and I apologize. The hyaluronidase breaks the corona radiata cells apart, and then acrosin, which is the other enzyme, there we go. Acrosin breaks down the zona pellucida. So the hyaluronidase breaks through the corona radiata in between the cells, and the acrosin breaks through the zona pellucida. And again, it takes dozens of sperms worth of acrosomal caps enzymes to break through the corona radiata and the zona pellucida for a sperm to get through to the oocyte. Okay, oocyte activation. Changes in the oocyte when it is fertilized. So when it is fertilized, the cell membrane of the sperm fuses with the cell membrane of the oocyte, and the genetic contents of the sperm are just injected into the egg. So it's not the whole sperm goes into the egg, it fuses. Anyway, the oocyte activations are the changes in the oocyte when fertilized. The trigger for activation is the fusion of the sperm and oocyte cell membranes. The process leads to an increased permeability in sodium. Increased permeability in sodium. And the entry of extra sodium, because sodium, like fools, rush in. Every time. Every time you open a sodium ion channel, sodium rushes in. So increased sodium permeability causes sodium to rush in. The influx of sodium causes, of course, the depolarization, and that causes a release of calcium plus from the smooth endoplasmic reticulum in the cytoplasm. And the influx of calcium causes three things, bullets. It hardens the zona pellucida, which does not allow a second sperm in. If you do get a second sperm in, it's called polyspermy. Poly means many. So it prevents polyspermy. And second bullet, it completes meiosis II to become an ovum. We mentioned this before, that the egg is released not as an egg, not as a full-fledged ovum, but as a secondary oocyte that has not finished meiosis II. And it will not finish meiosis II unless it is fertilized, and it's this calcium release from the SER because of the sodium, because of the sperm. It's a cycle. It's a thing. And that is what causes it to finish meiosis II and become a proper ovum. So now that it's fertilized, it has become an ovum, an egg. And third bullet, it makes proteins that it's going to need for development, which is a whole lot of cell reproduction. Okay, that was page 16. Let's go to page 17. We have 5 minutes left. Oh, I wonder if we can finish this page in 5 minutes. Okay, gestation is preggers. Gestation is pregnancy. Gustation is, what was gustation? Gestation is pregnancy, and we talked about gustation way back at the beginning of the semester. What was that? Taste. Gustation is taste, and gestation is pregnancy. And that's the kind of question that I love to put on the final. Take something from test 1, something from test 6, and they sound kind of alike, and they spell identical except for one letter. And you smoosh them into one question, and oh, so much fun. Although I usually tell you about those. We talked about Renin versus Renin. Okay. So let's talk about the trimesters. There are 3 trimesters. I feel like I'm on the wrong slide. That's because I was on the wrong slide. There are 3 trimesters. The first trimester, no, that's the next slide. I am missing a slide. Okay. 3 trimesters. The first trimester, you have the pre-embryo, embryo, and early fetal stages. And it begins all of the organ systems, which we're about to call organogenesis, but we haven't written that down yet, so we'll get there. The second trimester, you develop the organs and organ systems that you started in the first trimester. And then the third trimester, you gain weight. Get big enough to survive on the outside. And pregnancy is dated, and this is weird, from the first day of the last menstrual period, capital LMP, which is weird because that includes the 2 weeks before you had sex. And not everyone has regular cycles. So if you have irregular cycles, dating your pregnancy by the first day of the last menstrual period can be a week to a year off. So, that's what they go with. If you keep track of when you have sex, then that will help give a more accurate date. Okay. So the major stages of the first trimester. The first thing is cleavage, and this is when the cells divide. The zygote, which is the single-celled organism, divides to become a pre-embryo, which is a clump of cells. Second is implantation. Implantation is when the pre-embryo attaches to the uterine wall and embeds itself in the functional zone. 3 is placentation, which is growing a placenta. Placenta shun. Still weirds me out that T-I-O-N is shun. So is C-I-O-N, shun. How does that make sense? And embryogenesis is growing a viable embryo. And what percentage of conceptions survive the first trimester? Only 40%. So there are more conceptions that do not survive the first trimester than survive the first trimester. And many of those are lost before anyone knows that they're pregnant. So many of them are lost completely unawares. And for some people, they'll choose to not share the news of a pregnancy until they've passed the first trimester, so that they know there's a better chance that the pregnancy will continue, which makes sense because you don't want to tell everyone that you're pregnant and then lose the baby and then have to tell everyone that you're not pregnant. But it also doesn't make sense because then you're mourning a loss on your own and no one knows. And that's, you have to do what works for you, but consider, I don't know, everybody's got to do their own thing. So. The embryonic period is the 14 days from, the pre-embryonic period is 14 days from fertilization to implantation. So this is the pre-embryo. Also, this is a picture on the test. This is a picture on test 6. So know all of these terms. The lab terms we all need to know. We've got a zygote, fertilization, and then a zygote, and then the first cleavage. Each division is a cleavage, and this first cleavage makes 2 blastomeres, second cleavage makes 4 blastomeres. Once you've got a clump of cells, you use the term morula. Morula means raspberry? Not huckleberry, raspberry, I think. And that's a clump of cells. Oh, timer, hello. And then when this is the zona pellucida. When the zona pellucida is lost, that's called hatching. Is that on the note set? Absolutely no. Figure 29.2, cleavage and blastocyst formation, often a test picture. That's this picture. It's on the test. Oh, next page, page 18. It's what I just told you. Between the morula and blastocyst stages, the zona pellucida is shed in a process called hatching. You hatched, just like an egg. Well, it's fair. It's fair. An ectopic pregnancy is implantation of outside of the uterus. So standard place for implantation will be here, usually kind of higher up towards the back in the uterine wall. But sometimes it will implant in the uterine tubes, and that is not consistent with life. There is no chance for a baby in the pre-embryo in the uterine tubes to survive. In fact, even if they were to get the blood supply they need to grow, as they grow, they would rupture the uterine tubes and kill the mama. So tubal pregnancies, which is a type of ectopic pregnancy, must be surgically removed. And they cannot be reimplanted. Some politician somewhat recently decided to make it legally required to reimplant tubal pregnancies, and it's just like, so there's reality and then there's what the politicians say, and we're going with reality every time because you can deal with reality even when it's unpleasant, because reality can be changed, impacted. Wild conspiracy theories, not so much. So, the embryonic period is from implantation to 9 weeks, and here's a picture of implantation, and these are all lab terms except lacuna. So this is all information that you need to know. And hormones during this period. Do I have those typed in? I do not. Okay. Hormones during this period, we have lowercase h, capital C, capital G. And this is human, chorionic, because it's from the chorion, chorionic, gonadotropin. And we've talked about gonadotropin before. We had the two gonadotropins are FSH and LH. And this is a different gonadotropin. Gonadotropin? Gonadotropin. There we go. And it's from the chorion. And then from the corpus luteum, we have progesterone, and lower amounts of estrogen. estrogen from the corpus luteum until the end of the 2nd month, and then the placenta will be developed enough to pump out these hormones. It's progesterone and estrogen, but as you recall, progesterone is the main hormone that maintains the pregnancy. Unfortunately, progesterone is also the hormone that makes you nauseous, especially during the first trimester, although it can last the entire pregnancy. It's called morning sickness because it tends to be a little worse. It tends to be worse in the morning, but for some people it's all day long, all pregnancy long, which is frustrating because the hormone that makes you sick is the hormone that is primarily working to keep you pregnant. And so you're like, oh, I'm so sick, and the doctor's like, yay! Like, thanks. And then differentiation we talked about, and that involves changing the genetic activity of the cells. If the cell is going to be a bone cell, then you turn off the liver cell genes and turn on the bone cell genes. And this epigenetics. So if it's going to be a brain cell, you turn on the nerve cell genes and turn off the liver cell and bone cell genes. And so this is differentiation, and it happens slowly over time. And I'm past time, so we're going to call it here. And we will come back and talk about early embryogenesis. So this has been AMP2 Lecture Test 6, Video 9. And we did 15 through 18? Must have gone over time. Oh yeah, the timer went off, didn't it? Okay. 15 through 18. Thank you, as always, for tuning in. The education must go on. Dr. J out.